Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over briefly uronic acid pathway. And also I will be going over some of the important reactions in uronic acid pathway and applied clinical aspect related with uronic acid pathway, and that is essential Pentosuria. So let's get into the details. Now, uronic acid pathway, it is the formation of a molecule called glucuronic acid. You must have heard glucuronic acid, that is which is coming into the reaction as UDP-glucuronate. And note that this UDP-glucuronate molecule, which is an active form of glucuronate or glucuronic acid, it is used in the synthesis of mucopolysaccharides like glycosaminoglycans. You know, different types of glycosaminoglycans, they contain UDP-glucuronate, and it is also necessary for synthesis of glycoproteins. And UDP-glucuronate is also used in detoxification of certain drugs and chemicals. And UDP-glucuronate is used in conjugation of bilirubin, like conversion of unconjugated bilirubin into conjugated bilirubin, water insoluble bilirubin. How it is converted to water soluble bilirubin? It is by using UDP-glucuronate. And also UDP-glucuronate is used in steroid hormone biosynthesis. So there are varieties of uses of UDP-glucuronate, and uronic acid pathway is the one that will give glucuronate, which will be converted into UDP-glucuronate for all these functions. So, what happens to the unused glucuronate that is made in uronic acid pathway? Like unutilized glucuronic acid, it will be converted into xylulose 5-phosphate. And that xylulose 5-phosphate, as you know, it will enter into non-oxidative phase of pentose phosphate pathway, ultimately converted into fructose 6-phosphate, and fructose 6-phosphate will be converted into pyruvate, and pyruvate is converted into acetyl-CoA. Acetyl-CoA simply goes into TCA cycle for energy purpose. So this is what is briefly how uronic acid pathway will provide glucuronate, which is used for variety of biological reactions. So here is the pathway. Now the UDP-glucose, which is an active form of glucose entering into metabolic processes, UDP-glucose is converted into UDP-glucuronate. This will be done by UDP-glucose dehydrogenase enzyme, remember. So in this process, NAD is converted to NADH+, while glucose is converted into glucuronate. So glucose is oxidized into glucuronate here. Now the UDP-glucuronate, it can go into variety of biological reactions as I have shown you before. Now, UDP-glucuronate, it can be converted into glucuronate, deglucuronate, and deglucuronate, it's basically unoxidized, unutilized UDP-glucuronate. It can go into glucuronate formation, deglucuronate formation. Deglucuronate is converted to L-gluconate done by glucuronate reductase enzyme. And L-gluconate is converted to L-gulonolactone by aldolactonase enzyme. And L-gulonolactone in some of the and most of the plants and in some animals, L-gulonolactone is converted into L-ascorbic acid done by gulonolactone oxidase enzyme. And this is how ascorbic acid is synthesized in plants, like in citrus fruits, where ascorbic acid is at high concentration. So plants can synthesize L-ascorbic acid, and some animals can synthesize L-ascorbic acid because they will express gulonolactone oxidase enzyme. Whereas humans lack this gulonolactone oxidase. That's why L-ascorbic acid cannot be synthesized in humans. So what happens in humans? So it is L-gulonolactone, it will basically be converted into xylulose 5-phosphate, and xylulose 5-phosphate enters into pentose phosphate pathway and going into fructose 6-phosphate and then into pyruvate for energy purpose. So that's what happens. So the alternate fate of L-gulonate here, as I was saying, oxidative decarboxylation of L-gulonate occurs into L-xylulose. And this L-xylulose is reduced to xylitol by xylulose reductase enzyme, and xylitol is oxidized to D-xylulose and they are done by D-xylulose reductase enzyme, and D-xylulose is phosphorylated into xylulose 5-phosphate, and that xylulose 5-phosphate goes into pentose phosphate pathway. So ultimately what happens is L-gluconate is converted into xylulose 5-phosphate going into pentose phosphate pathway. That's how uronic acid pathway is connected with pentose phosphate pathway. Now let me show you that here. L-gluconate here, this L-gluconate ultimately by multiple steps will be converted into xylulose 5-phosphate, and this xylulose 5-phosphate going into pentose phosphate pathway, that is non-oxidative phase. Non-oxidative phase of pentose phosphate pathway, it will become fructose 6-phosphate, and fructose 6-phosphate going into pyruvate formation. So that's how a uronic acid pathway, this entire pathway, uronic acid pathway, is connected with pentose phosphate pathway. Okay, so a disorder that is associated with uronic acid pathway is essential pentosuria. This essential pentosuria is because of deficiency of 𝗏-xylulose reductase enzyme. You see where is the role of 𝗏-xylulose reductase here. It is the conversion of xylitol into xylulose done by xylulose reductase, and then as you see here, oxidative decarboxylation of L-gluconate to L-xylulose, and L-xylulose is converted to xylitol by L-xylulose reductase enzyme. So if there is a deficiency of this L-xylulose reductase enzyme, that will lead to decreased conversion of L-gluconate into xylulose 5-phosphate, and that may lead to elevation of L-xylulose in the cell, and that will be excreted out of our body in the urine. So there will be high levels of L-xylulose in the urine, and that is what is referred as essential pentosuria, because xylulose is a 5-carbon sugar molecule. So it's an asymptomatic condition, generally do not need any treatment. So that's what is essential pentosuria. You must be aware of this particular disease. It's an asymptomatic condition just like essential fructosuria. So deficient enzyme is L-xylulose reductase. Okay, so that is about uronic acid pathway. Thank you.